Good morning, students. Myself, Dr. Raghusudha, Head and Assistant Professor, Department of Microbiology, Government City College, Hyderabad, is here today with a topic on autoimmunity. Autoimmunity is breakdown of immune system's ability to discriminate between self and non-self. Condition in which structural or functional damage is produced by action of immunologically competent cells or antibodies against the normal components of the body. 5 to 7% of adults are affected, while two-thirds are only women. Greater than 40% of human diseases are of autoimmune origin. Autoimmunity is a condition characterized by the presence of serum antibodies and self reactive lymphocytes called T cells. It manifests sometimes when the body loses immune tolerance and mounts an abnormal immune attack, either with antibodies or T cells, against a person's own self antigens. In other words, the immune system of the body, like any complex multi component system, is subject to failure of some or all of its parts. When the system loses its sense of self and begins to attack cells and tissues of the body of its own, the result is autoimmunity. Autoimmune reactions can cause serious damage to cells, tissues, and organs and may result in diseases called autoimmune diseases. Three requirements to call its autoimmunity. First, presence of an immune reaction specific for some self antigen or self tissue. Evidence that such a reaction is not secondary to tissue damage but is of primary pathogenic significance, and absence of another well defined cause of the disease. All these three factors will make it confirm that that disorder is nothing but an autoimmune disease. Let's go through the history. Sergei Metelnikov in 1900 reported that some animals were able to form antibodies against their own spermatozoa. Paul Ehrlich in 1901 rejected the concept that an organism's immune system could attack the organism's own tissue, calling it horror autotoxicus. Julius Donath and Karl Landsteiner in 1904 reported autoantibodies can cause disease by showing that autoantibodies or hemolysis caused by paroxysmal cold hemoglobinuria. Ernst Widsky and Noel Rose in 1956 were able to induce an experimental autoimmune thyroiditis mediated by autoantibodies. Widsky in 1957 postulated in Widsky postulates modified later in 1994 for placing a disorder under autoimmune diseases. There are three conditions which were proposed or postulated by him, that the same conditions we have seen in the earlier slide. Direct evidence for transfer of pathogenic antibodies or pathogenic T cells. Indirect evidence based on development of autoimmune diseases in experimental animals. Conditional evidence from clinical clues from signs and symptoms of the patient. Before getting into the topic autoimmunity, as I've said you that it manifests when the body loses its immune tolerance. So let's see what is this tolerance. Normal immune system must differentiate between self and non-self. Immunological tolerance or immune tolerance is nothing but it is an unresponsiveness to an antigen that is induced by exposure of specific lymphocytes to that antigen. Failure of self-tolerance. Lack of responsiveness to an individual's own antigens underlies our ability to live in harmony with our cells and tissues. Autoimmunity results from the loss of the self tolerance. And this self tolerance or tolerance can be of two types: central tolerance and peripheral tolerance. Central tolerance means immature self reactive lymphocytes that recognize self antigens in generative or central lymphoid organs die by apoptosis or other fates. Peripheral tolerance means mature self reactive lymphocytes that recognize self antigens in the peripheral tissues are inactive, uh, or killed or deleted or suppressed. Immature T cells that recognize self antigens in the central lymphoid organs. Some are killed by apoptosis or negative selection or deletion in the thymus. Escapes killing and differentiation into regulatory T cells. In the B cell lineage, some are killed by apoptosis and some of the self reactive lymphocytes switch to new antigen receptors that are not self reactive, called receptor editing in the bone marrow. We have discussed about this negative selection or detection in thymus or deletion in thymus and apoptosis while discussing about proliferation of B cells and T cells. Kindly refer to that video in order to get a good understanding about this negative selection and apoptosis. Deletion of self-reactive T cells in the thymus. How are self-antigens exposed in the thymus? Many self-antigens are processed and presented by thymic antigen-presenting cells in association with the self-MHC. An immature T cell that encounters a self-antigen undergoes deletion or negative selection, and the T cells that complete this maturation are thereby depleted of self-reactive cells. AIRE or autoimmune regulator is a regulator of the gene transcription that stimulates thymic expression of many self antigens which are largely restricted to peripheral tissues. Mutation in this AIRE gene give rise to autoimmune polyendocrine syndrome, in which T cells specific for multiple self antigens escape deletion and attack tissues expressing in the self antigen. Peripheral tolerance. Self-reactive T cells that escape negative selection in the thymus can potentially wreak havoc unless they are deleted or effectively muzzled. Several mechanisms in the peripheral tissues that silence such potential autoreactive T cells are en- energy, or making them functionally inactive, or suppression by regulatory T lymphocytes, or deletion by activation induced cell death by activation of pro-apoptic membrane of BC1 family called BIM, fast ligand system, etc. Now let us see what are the factors influencing autoimmunity. Here we can see three factors: genes, then immune regulation, and environment. These are the three major factors which mainly influence the cause of autoimmunity. Let us see first one: genetic factors. There is increased expression of class 2 HLA antigens or tissues involved in autoimmunity. There is increased familial incidence of some forms of the autoimmune disorders. There is high incidence of autoimmune disease in twins, favoring genetic basis. Most human autoimmune diseases are associated with multiple genes. Genome-wide association studies are revealing genetic polymorphism associated with the autoimmune diseases. Here we can uh, understand that genetic factors play an important role in exhibiting this autoimmune disease, and these diseases are associated or linked with genetic polymorphism, which make them complicated in diagnosing. Next one is immunological factors. Polyclonal activation of B cells by stimuli such as infection with microorganisms and their products bypass T cell tolerance. Generation of self-reactive B cell clones bypassing T cell tolerance. Decrease of T suppressor and increase T helper cell activity leads to high levels of autoantibody production by B cells, contributing to autoimmunity. Fluctuation of anti-idiotype network control may cause failure of the mechanism of immune tolerance. Then sequestered self-antigens means the antigens which are not exposed while developing the immune system at the infant stage may act as foreign antigens if introduced into the circulation system later. So sequestered antigens, these are certain self antigens are present in closed systems and are not accessible to the immune apparatus. These are known as sequestered antigens or hidden antigens. Many self antigens are found in hidden locations, example, central nervous system, testes, eye consists of cornea. Cornea is also not exposed to the immune system. When these organs are damaged, then the hidden antigens are released and reaches the bloodstream. They encounter antigen sensitive cells and stimulate autoimmunity. The next factor is environmental factor. UV radiation causes cell death and may lead to the exposure of nuclear antigens which elicit pathologic immune response in lupus disease. Lupus is an autoimmune disorder. Smoking is another risk factor for rheumatoid arthritis, perhaps because it leads to chemical modifications of the self antigens and strong gender bias, with many of these diseases being more common in women than men owing to hormones and other mechanisms. So here we can see some of the very common autoimmune diseases. All of us have come across asthma. Asthma is a very commonly heard autoimmune disease, and the next common autoimmune disease is rheumatoid arthritis. And these are apart from these two, there are many other autoimmune diseases like Hashimoto's thyroiditis, systemic lupus erythematosus, then multiple sclerosis, celiac disease, and eczema and psoriasis. So these are some of the autoimmune diseases which we usually come across. How do we diagnose these autoimmune diseases? What kind of laboratory evidence is required for diagnosing the autoimmune disease? Presence and documentation of relevant autoantibodies in the serum, demonstration of T cell reactivity to self antigen, lymphocytic infiltration in the pathologic lesions, production of cytokines by helper T cells, example interferons and interleukin 4, evidence to support production of pathologic lesions in the tissue by transplacent transmission, transfer of an autoimmune disease to an experimental animal by administration of the autoantibodies. So these are the things which which are to be considered while diagnosing the autoimmune diseases. Criteria for diagnosis of autoimmune diseases, the clinical evidences is association of other autoimmune diseases, improved therapeutic response to immune suppressive agents, that is when immune suppressive drugs are being administered to the patient, the autoimmune disease is found to be reducing, the symptoms are to be subsiding, then family history of autoimmune disease. So, what is the treatment? Treatment for this autoimmune diseases. The current therapies provide relief by non-specific suppression of the immune system. Some agents like cyclosporin A are somewhat selective in that they inhibit only antigen activated T cells. More directed approaches are still at experimental level. Some of these are T cells specific for concerned antigen are injected into individuals to immunize them against these T cells, which is also known as T cell vaccination. So that is one kind of treatment for autoimmune diseases, wherein the T cells which are specific to the concerned antigen, the antigen which is the causative for the autoimmune disease, to that antigen T cells are being injected, and this is known as T cell vaccination. The other treatment is a synthetic peptide differing by only one amino acid from the autoantigenic peptide may be used as a therapy. The synthetic peptide competes with the autoantigenic peptide of major histoability complex molecules. And one more method for treatment of autoimmune diseases, monoclonal antibodies may be directed against CD4 cells. Then the alpha subunit of interleukin 2 receptors, which is expressed only by the antigen activated T helper cells. Then specific T cell receptors and the specific allelic variants of the MHC molecule that is associated with the autoimmune disease. And one more method for treatment of autoimmune diseases is oral administration of the concerned autoantigen, which induce tolerance to the antigen. Thank you.